Hey, thanks for the kind introduction. Let me quickly put on the laser pointer and then I think I'm ready to go. Okay, so animals rely on olfaction to find food, attract mates, and avoid predators. And this requires the identification of the same odor across a range of intensities, but also it requires discrimination of odor intensities themselves. And this is especially important during tasks like navigation, for example. When we look at the olfactory system, odor molecules activate the peripheral olfactory neurons that project to mitral cells in the olfactory bulb, the initial odor processing station in the central nervous system. And these mitral cells then in turn project to several areas, including the piriform cortex, the primary olfactory cortex, this large portion here. So a recent paper from Kevin Bowling and Franks shows that odor-evoked activity in the olfactory bulb comes in two phases, and this is known for a long time. And it's shown here at the mean firing rate of neurons recorded from the olfactory bulb. They show that there's an initial phase that is largely invariant for different odor intensities. And then there's a late phase that is changing more dramatically if they present different odor intensities, shown here from yellow to red. In contrast, the piriform cortex, so the primary olfactory cortex, only responds to this initial phase. And they show that recurrent connectivity in the piriform cortex suppresses the activity during the sustained phase. And we conclude from this that the piriform cortex represents odors invariant to intensity, allowing to identify the same odor across a large range of concentrations. So, but odor intensity is also important. So where's odor intensity processed? There are several other areas that receive input from the olfactory bulb. So we wondered, since odor intensity is especially important during navigation, if you, for example, want to find food or something, we wondered if the direct projections from the olfactory bulb to the lateral entorhinal cortex may transmit this information about odor intensity to the entorhinal hippocampal network. So to test this hypothesis, we performed extracellular recordings from awake head-fixed mice simultaneously from the olfactory bulb, piriform cortex, and LEC, or from the olfactory bulb, LEC and hippocampus. We used a pressure sensor to record the respiration of the mice and give a closed-loop odor stimulation triggered by the exhalation of the mice. So when we look at the PSDHs that we give from this exhalation-triggered odor stimulation, they look quite blurry in time, actually. So we realigned the trials to the first inhalation that is coming in after odor onset. And when we do that, we see that odor-evoked activity is actually quite precisely timed for this example neurons from the lateral entorhinal cortex. So we looked at the average activity for all units in these areas. And as described before, we see this initial phase, and if we give odors shown here in blue, at about within the first 100 milliseconds after inhalation in the olfactory bulb, and then this late phase, the sustained phase that is coming later on from about 100 to 400 milliseconds. And again, we saw that the piriform cortex only responds to the initial phase. To our surprise, however, the initial phase of odor-evoked activity also strongly dominated in the lateral entorhinal cortex, as well as in dentate, gyrus, and CA1 of the hippocampus. So we wondered, is the LEC actually important for the discrimination of odor intensities? We decided to test this using a two alternative forced choice task in mice. We used get-to-agree mice to express gyrotopsin selectively in interneurons, which allows us to transiently silence the LEC. First, we trained mice to discriminate between two odors and reported choice by licking left for odor A or right for odor B. And similar to the electrophysiological recordings, odor representation was triggered by exhalation. To our surprise, simple odor discrimination was impaired when we silenced the LEC bilaterally, which is kind of unexpected because it was supposed to mainly depend on the piriform cortex. When we looked at the timing of the LEC responses of error trials and correct trials, we see that already at about 200 milliseconds, the mice are able to distinguish correct choice and perform above chance. So that indicates that this fast component that this initial fast component is important. And again, here this timing was aligned to the first inhalation after odor onset. Now, when we trained mice to discriminate intensities, they were also able to do the task, but again, their performance dropped to chance when we silenced the LEC. And again, they were able to discriminate the two intensities already about 200 milliseconds after inhalation. So sometimes they respond later. We don't force them here to respond fast, but that's when the correct trials and error trials basically are separated from each other. So this shows that the LEC is critical for the discrimination of odor intensities, and the timing suggests that the initial phase of odor-evoked activity does not only contain information about odor identity, but also about odor intensity. So we decided to look at the local network in the LEC in more detail to understand what is going on. We focused on layer 2 of the LEC, which is the main input layer for olfactory information and the main output layer to the hippocampus. There are two different cell types in layer 2 of the LEC, fan cells and pyramidal cells. And fan cells express reelin and are located in layer 2A, which you can see here, whereas pyramidal cells express calbildin and are located a little bit further away from the surface in layer 2B. We identified a new driver line, Netrin G1, mouse to selectively express target expression in fan cells. And expression in pyramidal cells can be selectively targeted with calbildin driver line. We used Netrin G1 mouse line to express TD tomato in fan cells of the LEC and looked at their projections to the hippocampus. And we found that fan cells project to the outer molecular layer of the dentate gyrus by the lateral preferentially path. In contrast, when we use calbildin mice, we see that pyramidal cells directly project to the stratum maculoso molecular of CA1. So fan cells project to the first station of the trisynaptic hippocampal loop, the dentate gyrus, whereas pyramidal cells project to CA1, the last station of the trisynaptic loop, where the two pathways actually converge again. We patched fan cells in LEC slices and filled them with biocytin to look at their anatomical and physiological properties. And while we found subtle differences in their physiology, actually the most dramatic difference was the anatomy. The fan cells show very distinct dendritic morphology than pyramidal cells and span a large area at cortical surface where the direct projections from the olfactory bulb arrive. So we performed paired recordings of fan cells and pyramidal cells and optogenetically stimulated incoming axons from the olfactory bulb to compare the direct inputs of the two cell types. We found that fan cells receive much stronger input compared to pyramidal cells from the olfactory bulb. Minimal stimulation of single axonal fibers showed no difference between fan cells and pyramidal cells, indicating that there is no difference in synaptic strength that two cell types receive, but in the number of inputs. When we stimulated axons coming in from the piriform cortex, again, we found that fan cells receive stronger input than pyramidal cells. However, the difference is less dramatic than for the input from the olfactory bulb. So, but what does this mean for odor-evoked activity? To address this question, we performed extracellular recordings from the LEC in awake head-fixed mice, expressing generodopsin in fan cells or pyramidal cells, which allows us to identify them by phototag. And we looked at odor-evoked activity of individual units, and each line here represents the mean activity of one unit to different odors. We saw that the initial phase of odor-evoked activity within the first 100 milliseconds, dominating the response in fan cells as well as pyramidal cells. This becomes even clearer when we look at the average odor-evoked activity for fan and pyramidal cells. So again, fan cells respond to the initial phase, not to the late phase, and the same is true for pyramidal cells. So when we looked at the precise timing of the initial phase, so this is an expansion of this part here, basically overlaid, we saw that fan cells respond before pyramidal cells, which is consistent with the strong direct input from the olfactory bulb. And it's interesting because they basically activating the local inhibitory network, maybe suppressing further activity. So, but what about odor intensity? To test this, we presented odors at 4 different concentrations shown here by different color situations from low concentration to high concentration. And again, we only look at this initial phase where we actually saw clear responses. And we see that fan cells increase their firing rate with increasing odor intensity already during this initial phase. Pyramidal cells also increase their firing rate, but additionally, they responded faster with increasing odor intensity, indicating an additional temporal code. So peak times of the odor responses at different intensities are summarized here and show the faster response of pyramidal cells, which is also there, but way less dramatic in fan cells. And this results in a decreasing time difference of fan cell to pyramidal cell activity with increasing odor intensity. So that's interesting because for downstream area, a temporal code only makes sense if there's a reference signal, since it does not have the information when the inhalation has happened. So our data suggests that fan cells might provide this reference signal, and the timing of pyramidal cell activity relative to the fan cell activity can be used by a downstream area to read out odor intensity, which might happen in CA1 where the two pathways actually converge again. So to summarize, I showed you today that mice can rapidly discriminate odor intensity, and this requires the lateral entorhinal cortex, and especially the initial phase of odor response in the lateral entorhinal cortex. LEC fan cells receive more direct input from the olfactory bulb and respond before pyramidal cells. Fan and pyramidal cells increase their firing rate with odor intensity, and pyramidal cells additionally respond faster as odor intensity increases. And this timing difference between fan and pyramidal cells could encode for odor intensity. Yeah. And this, I want to thank to the lab, everyone in the lab, and the funding, of course, and thanks for watching.
Thank you so much for that lovely talk. While we're waiting for questions to come in, we have a few minutes for questions. I had a question which was related to the fact that the fan cells and pyramidal cells project to different regions of the hippocampus. Do you have speculations as to what then would happen to those hippocampal representations, that you would get, say, fan cells that have more olfactory inputs coming in projecting to dentate versus pyramidal cells to CA1?
That's something that we want to look into next, basically what is going on in the hippocampus. So one idea would be that the fan cells that get a more direct input from the olfactory bulb, it has recently been shown that the odor representations in the olfactory bulb are very stable, whereas odor representations in the piriform cortex change over days quite a bit. So maybe the direct projections onto the fan cells and then their projections to the dentate cells also result in a more stable odor representation over days in the dentate gyrus, whereas the one at the CA1 might be more varying with time. But yeah, that's what we were planning to look into.
Kind of as a follow-up, have you thought about looking into whether dentate is more involved in odor discrimination, similarly to how it's more involved in, or hypothesized to be more involved in, pattern separation?
It's a good question. We didn't look into this yet, but it would be interesting to do for sure. Yeah.
Okay, well, I'm sure that we will have more questions.